This is the review session for the midterm exam 2, and its purpose is to narrow down the topics that you need to study for preparing the upcoming exam. But that doesn't necessarily mean that you are going to get covered 100% by looking at this review. You still have to go back to each lecture, revisit all the details about that, and have to memorize the things that I pointed out during the lecture. But this review session, again, will definitely help you to focus on certain topics. So the first thing that we discussed was the protein, especially part 2, focusing on their structures. So we talked about the different levels of a protein structure from primary to quaternary, and you should be able to tell what are the differences between those structural levels. And especially this certain type of interactions are important for the forming that protein structure, such as disulfide bond. We also talked about the phylogenetic tree, what it means along the evolution, as well as what these branch points mean, and the distance between the different species, and so on. We also talked about the protein sequencing, their methods such as Edman degradation, as well as mass spectrometry too. We also discussed the chromatography, which is for determining the amino acid composition, and different type of such chromatography, like the size exclusion, ion exchange, as well as affinity chromatography. This is the process of Edman degradation that we talked in details. In mass spec 2, we talked about how it works and what it detects. We also talked about protein purification, the purpose of that, as well as all different type of methods. And several methods are used to evaluate the protein purification, such as SDS-PAGE, which uses electrophoresis, as well as Western blotting or the ELISA too. And you should know how they work. In protein extraction 2, what are the steps and what kind of methods can be used? And in protein part 3, we discussed mainly about the protein structure, the rules that governing the 3D structure of the protein, as well as the different type of interactions between the residues among such polypeptides. So again, these are the structural elements from primary to quaternary, and you should also know about the domain as well as the supersecondary structure. And we've discussed quite intensively the phi and psi angles within the polypeptide and their steric conformation that is either allowed or crashed. And we've discussed the Ramachandran plot that shows the different conformation, the secondary structure of the polypeptides based on their phi and psi angles. And for beta sheets, there are two different structures, the antiparallel or the parallel. And the linker has a different property for such different subtypes of the beta sheet structures. And we talked about the motifs, and you should know what they are. And the protein domains too, their definition as well as their relative size. And again, the tertiary structure of a protein, how they are formed. And to determine the 3D protein structure, you can use different methods such as X-ray crystallography or NMR. And in protein part 4, we talked about the quaternary structure of the protein as well as their functions, especially the enzymatic activities. Here we talked about the protein folding, how it works, and the native state of the protein as well as non-native too. And energetically or thermodynamically, how the protein can achieve this native state by following the energy landscape. And we discussed the E. coli GRO-EL, GRO-ES complex that uses the ATP. And also, we talked really a lot about this myoglobin and hemoglobin, their differences in their structure, their quaternary structure especially, as well as their functions. And we discussed the binding curve for the oxygen and the difference between the myoglobin and hemoglobin and why what actually makes that difference too. And we discussed different factors affecting the oxygen affinity of hemoglobin as well as myoglobin. And we've also covered the sickle cell anemia, what causes such disease. And for 2 lectures about carbohydrates and glycoconjugates, we talked about the smaller units of carbohydrates as well as their polymers, and also some glycoproteins. You should know and memorize the monosaccharides and their structures, their types, different isomers, as well as their numbering, and their Fischer projection for different orientations. And also difference between the Fischer projection and the Haworth projection. And also there are different conformations such as alpha and beta for those sugar molecules. Glycosidic bonds are really important for making the oligosaccharides or polysaccharides from the monosaccharides. And you should be able to tell the difference between reducing and non-reducing end. Here in part 2, we mostly focused on the polysaccharides, their structures and their functions, and their different types that can be seen in different species, and their structures, especially if they make branches or not too. And we've discussed the glycoproteins, especially with the different linkages, and is that O-linked or N-linked, and what are the difference in terms of their structure and their function too. And again, you should memorize the structure of glucose, lactose, and so on of the monosaccharides, as well as their cyclic forms, and also some features and the structures of polysaccharides too. And for 2 lectures about lipids, membranes, and membrane transport, we focused on the different structures and component types of lipids, and the biological membranes, how they are formed, and what are the functions of them, what the controls are, as well as the membrane transport proteins that are stuck in the membrane to basically regulate the biological functions. These are the different classification of lipids and that you should know their structures, their differences in their functions, and so on. And among different types of lipids, some subtypes of lipids are a component of the membranes, and so they play a critical role in forming such biological membranes. And steroids and their precursor cholesterol is another type of lipids that have a peculiar structure. The lipids can make different structures such as micelle, bilayer, and liposomes, and that depends on their individual units. And we've discussed a fluid mosaic model, which suggests a different type of membrane proteins as well as in their dynamics. In part 2, we talked about different membrane transport proteins. The first one was integral protein, as well as we talked about peripheral membrane protein, lipid-anchored protein, glycoproteins, and so on. And membrane fluxes are driven by these different type of forces. And you should understand the permeability, how it is driven, and also the property of that. And different substances have different permeability through the biological membrane. For example, hydrophobic substances have high permeability because of its similar hydrophobicity as the lipid bilayer. And we've discussed the transmembrane protein, and especially talked about the difference between these carrier proteins and channel proteins. And for the active transport through the membrane, there are 3 major mechanisms that you should know how they exactly work. And there are several stimuli that make the different type of channel protein work. And so far, through this review session, if there are any topics that you feel you're unfamiliar with, or you think that you studied not too well, then you should go back to the lectures, the pre-recorded lectures, as well as the textbook, to make sure you understand everything about that.